Basics of Medical Biochemistry. Chapter 8. Biochemistry of the Gastrointestinal Tract. 8.1 Introduction. Digestion is a biochemical process of hydrolysis during which large and complex food particles are converted into small and diffusible particles so that they can be absorbed easily. It is carried out by various digestive juices, particularly enzymes at suitable pH. Digestion is important to break down food into absorbable nutrients, which the body uses for energy, growth, and cell repair. There are two aspects of the mechanism of digestion. Chemical digestion uses enzymes to break down food. Mechanical digestion involves the physical movement of the tongue, teeth, and muscles of various parts of the gastrointestinal tract. The digestive system in humans is composed of a long muscular tube called the gastrointestinal tract and accessory digestive glands. The gastrointestinal tract consists of the oral cavity, pharynx, esophagus, stomach, small intestine, large intestine, rectum, and anal canal. The accessory organs include the teeth, tongue, salivary glands, pancreas, liver, and gallbladder. Absorption is the process of translocation of nutrients from the gastrointestinal tract into the bloodstream. Transport is the process of movement of absorbed nutrients throughout the body via the circulatory and lymphatic systems. At the cellular level, it encompasses the transfer of substances across the cell membrane. Elimination/egestion is the excretion of undigested and unabsorbed food through the feces, stool, via the act of defecation. 8.2 Digestive juices. Digestion of major foodstuffs is an orderly process involving the action of a variety of digestive enzymes. These enzymes are found in various secretions of digestive organs. Main digestive juices include saliva, gastric juice, pancreatic juice, and intestinal juice. The process of digestion and absorption is also aided by the substances like hydrochloric acid secreted by the stomach and bile by the liver. 8.2.1 Saliva. Saliva is a mixture of secretions from three pairs of salivary glands. 1. The parotid glands. These are the largest salivary glands and secrete watery fluids. 2. The submandibular glands. Their secretion is serous, watery, and mucus type. Their secretion is viscous because it contains a higher concentration of proteins. 3. The sublingual glands. These are the smallest of the three salivary glands. Like submandibular glands, their secretion is also serous, watery, and mucus type. In addition to the above three types, there are numerous small salivary glands in the buccal mucosa located throughout the oral cavity. Their secretion is of mucoid type. Those in the tongue secrete lingual lipase. Normally, a moderate amount of saliva is continuously secreted to keep the mucous membrane moist and to lubricate the movements of the tongue and lips during speech. About 20 ml of saliva is secreted per hour. The amount of saliva secreted per day varies considerably, ranging from 0.5 to 1.5 liters daily. Salivary secretion is entirely under nervous control. Salivation is increased by smell, sight, thought, touch, or sound of food preparations. Composition of saliva. The composition of saliva is quite variable. On average, its water content is 99.5% and solid content is 0.5%. Of the solids, inorganic constituents are 40% and organic constituents comprise 60%. Saliva is slightly acidic, pH 6.4 to 6.9. Inorganic constituents. The main inorganic constituents include cations such as sodium, potassium, calcium, and magnesium, and anions such as sulfates, chlorides, phosphates, and bicarbonates. Thiocyanate ions are also present, especially in smokers. Both electrolyte concentration and osmolality vary with secretory flow rate, but generally, in comparison to plasma, saliva is hypotonic and contains higher concentrations of K+ and bicarbonate, HCO3, and low concentrations of Na+ and Cl. 2 Organic constituents. The chief organic constituents of saliva are mucin and enzymes. Mucin is a mucoprotein. Approximately 1/3 of mucin is carbohydrate. The lubricating property of saliva is mainly due to its mucin content. The main enzymes in human saliva are amylase and lysozyme. Amylase is an amylolytic enzyme whereas lysozyme is antibacterial. Ebner's glands found on the dorsum of the tongue secrete lingual lipase. Other organic constituents are urea, cholesterol, immunoglobulins such as IgA and some vitamins. The saliva of some persons also contains specific blood group substances, therefore, these individuals are also called secretors. Figure 8.1 Composition of saliva. As a result of sympathetic stimulation to salivary glands, the arteries of the glands get constricted and the blood flow is much reduced. A small amount of viscous saliva containing a high percentage of solids appears. As a result of parasympathetic stimulation to the parotid gland, a copious flow of watery saliva is noticed, which is rich in ptyalin. Nausea and intestinal irritation also stimulate salivation. Fear, fatigue, sleep, and dehydration inhibit salivation. Acetylcholine, pilocarpine, adrenaline, and ephedrine all cause increased secretion of saliva. Atropine has the opposite effect. Functions of saliva. Articulation, saliva moistens the mouth and facilitates speech. Buffer, it contains HCO3 and mucin buffers to neutralize ingested acid or alkalis to keep the pH of the mouth at 7. This pH resists demineralization and teeth do not lose their calcium and thus, remain healthy and strong. Modifies food temperature, saliva helps in cooling hot food. Cleaning and antibacterial actions, it contains lysozymes, that destroy bacteria, and antibodies, IgA, against any antigen in the oral cavity. Deglutition, swallowing saliva helps in mastication and swallowing by lubricating the food. Digestion, salivary amylase breaks down starch to dextrin and then to maltose. Lingual lipase initiates the digestion of triacylglycerols that continues up to the stomach. Excretion, chemicals such as potassium, iodide, fluoride, mercury, lead, bismuth and urea are excreted in saliva in severe kidney disease, whereas sugar in severe diabetes and the virus of rabies in hydrophobia. Taste sensation, it facilitates the sensation of taste by dissolving food materials. Water regulation, as dryness of the mouth and decreased salivation, develop the feeling of thirst. Heat regulation, in panting animals with no sweat glands, example dogs and cats. 8.2.2 Gastric juice. Gastric juice is nearly a colorless, strongly acidic liquid secreted by the gastric glands. Composition of gastric juice, it is 99.5% water and 0.5% solid contents. Its active food-dissolving ingredients are the digestive enzymes pepsin and renin, which break down proteins, and hydrochloric acid, HCl. Gastric juice also contains mucus to protect the stomach lining from being dissolved by the HCl and pepsin. Figure 8.2 Composition of gastric juice. Organic constituents, pepsin, the proteolytic enzyme, mucin, intrinsic factor, a mucoprotein, necessary for the absorption of vitamin B12, gastric renin, not present in human adults, and other gastric enzymes, lysozyme, carbonic anhydrase, etc. 2 Inorganic constituents, free hydrochloric acid, lactic acid and other fermenting acids, sulfates, chlorides, phosphates of sodium, potassium, calcium, magnesium, bicarbonates, etc. Functions of the gastric juice. Pepsin initiates protein digestion. It is secreted as pepsinogens, inactive proenzyme, by chief cells. It is activated to pepsin by HCl. It is most effective at pH 1.5 to 2.0. It converts proteins into simpler, more easily absorbed substances. Its action is aided by hydrochloric acid, which provides the acid environment in which pepsin is most effective. Renin aids the digestion of milk proteins. Coagulation of milk in infants is the action of renin. Gastric lipase results in the hydrolysis of triacylglycerols to diacylglycerol and free fatty acids. Mucus secreted by the gastric glands helps protect the stomach lining from chemical injury by the action of gastric juice. Mucus also prevents mechanical injury to the stomach by lubricating the contents. Figure 8.3 Structure of gastric gland, cell type, and secretory products. Role of HCl. it is secreted by parietal cells also called oxyntic cells of the gastric gland. Acid sterilization, killing of bacteria and other microbes that make entry through food. Dissolve hard and bony particles of food. Activation of pepsinogen. Promotion of secretin secretion. Assists in the absorption of iron and calcium. Some other functions of the stomach include temporary storage of food, allowing time for digestive enzymes to complete their actions. Mechanical breakdown of the food. Absorption of water, some lipids and certain drugs. Production of the intrinsic factor for the absorption of vitamin B12. Figure 8.4 Mechanism of secretion of gastric juice, source, Elsevier Ltd., 2006. 8.2.3 Pancreatic juice, exocrine part of the pancreas secretes pancreatic juice consisting of enzymes and aqueous alkaline, NaHCO3, secretions. Composition, it is composed of 99.5% water and 0.5% solid contents. It is rich in HCO3 content being 5 times higher than plasma. Other inorganic constituents include sodium, calcium, potassium, magnesium, chloride, phosphate, and sulfate. Main organic constituents are proteolytic enzymes, trypsinogen, chymotrypsinogen, proteases, procarboxypeptidase, lipolytic enzymes, pancreatic lipase, cholipase, phospholipase A2, amylase, cholesterol esterase, RNase, DNase and alkaline phosphatase. It also contains albumin, globulin, etc. Figure 8.5 Composition of pancreatic juice. Bicarbonate ion production in the pancreas, it occurs in the following steps. 1. Co2 diffuses to the interior of the ductal cells from blood and combines with H2O bicarbonic anhydrase to form carbonic acid, H2CO3, which will then dissociate into HCO3 and H+. The HCO3 is actively transported into the lumen. HCO3 is exchanged for Cl. 2. The H+ formed from the dissociated H2CO3 is exchanged for Na+ ions by active transport through blood, which will diffuse or actively be transported to the lumen to neutralize the negative charges of HCO3. 3. The movement of HCO3 and Na+ ions to the lumen causes an osmotic gradient, that ultimately causes water to move from the blood to ductal cells of the pancreas, producing eventually the HCO3 solution. Figure 8.6 Production of bicarbonate ion in the pancreas. Phases of pancreatic secretion. The pancreatic secretion occurs in the following 3 phases. I. The cephalic phase contributes approximately 25% to the pancreatic response and is mediated by the vagus nerve. The stimulants are sight, smell, taste, and eating of food. The vagal effect is mediated not only by acetylcholine but also by other neurotransmitters like vasoactive intestinal polypeptide, VIP, and gastrin-releasing peptide, GRP. 2. The gastric phase contributes 10% to the response, and also via vagal innervation, mainly through stomach distension as it fills with food. 3. The intestinal phase, 50 to 75%, is mediated by secretin and cholecystokinin. The entry of acidic chyme into the small intestine stimulates the release of secretin and cholecystokinin, CCK, hormones. The CCK released can activate vagal afferent neurons that carry the signal to the dorsal vagal complex where the sensory information is integrated and vagal efferent nerves are activated. Vagal efferent synapse with neurons in the pancreatic ganglia. In turn, via the neurotransmitters, acetylcholine, GRP, and VIP effector neurons in the pancreatic ganglia activate secretion by pancreatic acinar cells. In addition to activating the neural pathway, CCK enters the general circulation and may act on the pancreatic acinar cells to cause secretion. The pancreas produces a specific peptide known as a pancreatic polypeptide, PP, which gives negative feedback on pancreatic secretion, i.e., it inhibits enzyme secretion. PP is released in response to vagal nerve stimulation. Figure 8.7 Regulation of pancreatic secretions. Functions of the pancreatic juice. The main function of pancreatic juice is to digest carbohydrates, proteins, and lipids by respective enzymes. Neutralization of acid chyme is another important function of pancreatic juice because of its high HCO3 content. The concomitant action of HCO3 and bile makes the pH of duodenal contents between 6.0 to 7.0, which is necessary for the actions of digestive enzymes here. 1. Pancreatic proteolytic enzymes. The protein-digesting enzymes are secreted as inactive proenzymes, zymogens. In the duodenum, zymogens are converted to active enzymes when enteropeptidase, produced by intestinal mucosa, converts trypsinogen to trypsin. Trypsin, in turn, activates the other pancreatic zymogens. I-trypsin, it is the main enzyme that catalyzes the activation of all other pancreatic enzymes such as chymotrypsinogen, procarboxypeptidase, and proelastase. Trypsin is an endopeptidase that hydrolyzes the centrally situated peptide bonds formed by basic amino acids, example lysine and arginine. Its optimum pH is 8.0 to 9.0. 2. Chymotrypsin, it is also an endopeptidase that hydrolyzes the central peptide bonds formed by aromatic amino acids. 3. Elastase, it hydrolyzes peptide bonds formed by small amino acids, example glycine, alanine, and serine. 4. Collagenase, it catalyzes the hydrolysis of collagen. V. Carboxypeptidase, it is an exopeptidase. It hydrolyzes peptides in the bonds adjacent to the free COOH group of the polypeptide chain, releasing each time a single free amino acid. 2. Pancreatic lipase, it is the most important lipase in triacylglycerol digestion. It digests the primary ester bonds, positions 1 and 3, hydrolysing them into fatty acids and 2-monoacylglycerol. A protein cholipase is also present in pancreatic juice. It is required to stabilize the lipase to its triacylglycerol substrate. 3. Pancreatic alpha amylase, its optimum pH is 7.1 and it is activated by chloride ions. It can act on cooked and uncooked starch and hydrolyzes them into maltose and isomaltose. 4. Cholesterol ester hydrolase, it hydrolyzes the cholesterol ester to cholesterol and free fatty acid. 5. Phospholipase A2, it acts on lecithin and cephalin, and converts them to lysolecithin and lysocephalin respectively. 8.2.4 Intestinal juice, it is a clear to pale yellow watery secretion from the glands lining the walls of the small intestine. These glands include 1. Acinotubular duodenal glands, Brunner's glands. They are located in the first few centimeters of the duodenum, where the pancreatic and bile juices pour into the duodenum. These glands produce a slightly alkaline, highly viscous fluid containing mucins. The function of the mucus is to protect the duodenal wall from digestion by the gastric juice. 2. Simple tubular intestinal glands, crypts of Lieberkühn. These are located on the entire surface of the small intestine as small pits called crypts of Lieberkühn. They secrete a fluid that is similar to the ECF but has a slightly alkaline pH in the range of 7.5 to 8.0. Composition of the intestinal juice, it is composed of 98 to 99% water and 1 to 2% solid constituents. Its main inorganic constituent is HCO3. Other inorganic constituents include sodium, calcium, potassium, magnesium, chloride, phosphate, and sulfate. Main organic constituents are mucus, enzymes, immunoglobulin A, IgA, intrinsic factor, and shed cells from the mucosa of the small intestine. Enzymes that are secreted in succus entericus are enteropeptidase and intestinal amylase. Some enzymes that are not secreted but remain within the surface of epithelial cells are oligosaccharidases, peptidases, phospholipases and nucleotidases, RNase and DNase. The pH of intestinal juice is 7.6 to 8.2. Regulation of secretion of intestinal juice. Intestinal secretion is regulated by both nervous and hormonal mechanisms. Nervous regulation, stimulation of parasympathetic nerves increases the secretion while stimulation by sympathetic nerves decreases secretion. However, the local nervous reflexes play an important role in increasing the secretion of intestinal juice. When chyme enters the small intestine, the mucosa is stimulated by tactile stimuli or irritation. It causes the development of local nervous reflexes, which stimulate the glands of the intestine. 2. Hormonal regulation. When chyme enters the small intestine, intestinal mucosa secretes secretin and cholecystokinin that promote the secretion of intestinal juice. Functions of intestinal juice. Bicarbonate helps in the neutralization of the acidity of chyme. The function of mucin is to protect the duodenal wall from digestion by gastric juice. It also buffers the HCl present in the chyme. Enteropeptidase, enterokinase, converts trypsinogen to trypsin. Intestinal amylase helps in the digestion of polysaccharides. A small amount of intrinsic factor is also found in the intestinal juice formed in the duodenum. It helps in the absorption of vitamin B12. IgA present in intestinal juice gives protection against microbes. Figure 8.8 Composition of intestinal juice, succus entericus. 8.2.5 Bile juice. Bile or gall is a yellow-green fluid secreted by the liver. Bile is secreted by the liver epithelial cells, called hepatocytes, and by epithelial cells lining the bile ducts called ductal cells. About 250-1000 ml of bile is secreted daily. Bile is drained from the liver to the gall bladder where it is concentrated and stored. The lining columnar epithelium of the gall bladder reabsorbs water and salts and adds mucus to it. The gall bladder releases the bile into the duodenum via the common bile duct after meals. Though it does not contain any digestive enzyme, it plays an important role in digestion and absorption of fats and fat-soluble vitamins due to its bile salt contents, particularly through the process of emulsification. It also serves to excrete many unnecessary and harmful substances, example bile pigments, metals, drugs, toxins, etc. Figure 8.9 Composition of bile juice. Composition of bile. Bile contains 97.6% water and 2.4% solid substances. The main constituents of the bile are given below. Bile salts. The main bile salts are sodium glycocholate and sodium taurocholate. Liver cells unite cholic acid with glycine and taurine to form sodium glycocholate and sodium taurocholate respectively. They activate pancreatic lipase, emulsify fat, form water-soluble complexes with cholesterol and fat-soluble vitamins, thus promoting their absorption, and stimulate liver cells to excrete bile, choleretic action. The total amount of bile salts is smaller than what is needed daily to promote lipid digestion and absorption. The enterohepatic circulation allows the recycling of bile salts. The body can recycle the same bile salts. Bile pigments. The chief bile pigment in human bile is bilirubin. The bile of herbivores contains appreciable quantities of biliverdin. Lipids. The major lipids are cholesterol and lecithin. Triacylglycerols and fatty acids are also present. Cholesterol protects gallbladder membranes from the irritating and harmful effects of bile acids and bile salts. Electrolytes. The most abundant electrolytes are sodium and bicarbonate ions. Proteins. The main proteins in bile are mucin and nucleoproteins. Table 8.1 Difference between hepatic bile and gallbladder bile. Component/characteristic feature. Hepatic bile. Gallbladder bile. Water. 97.5%. 85%. Solids. 2.5%. 15%. Proteins. 0.5%. 5%. Bile salts. 0.5%. 5%. Bile pigments. 0.3%. 3%. Lipids. 0.3%. 1%. Inorganic salts. 0.9%. 1%. Specific gravity. 1.010. 1.040. pH. 7.8-8.6. 6.5-6.9. Regulation of bile secretion. The secretion of bile is regulated by nervous and humoral factors. Nervous control is achieved through reflexes. The sight or smell of food provokes contraction of the gallbladder with the relaxation of the sphincter of Oddi. The taste of food also leads to reflex concentration of the gallbladder and relaxation of the sphincter of Oddi. Humoral control is mediated by a hormone from duodenal mucosa called cholecystokinin. The presence of fat in the duodenum stimulates the production of bile. Figure 8.10 Regulation of bile secretion. Source: https://medicoapps.org. Functions of the bile. 1. Fat emulsification is the process by which fat globules are separated into smaller pieces by the detergent action of bile salts supported by lecithin. Bile salts act as a surfactant and help to emulsify the fats. Bile salt anions have a hydrophilic site and a hydrophobic site and therefore tend to aggregate around droplets of fat to form micelles, with the hydrophobic sites towards the fat and hydrophilic towards the periphery. The hydrophilic sites are positively charged due to the lecithin, and this charge prevents fat droplets coated with bile from reaggregating into larger fat particles. 2. Bile serves as the route of excretion for bilirubin. 3. The alkaline bile also has the function of neutralizing any excess stomach acid. 4. The bile facilitates the intestinal absorption of fat-soluble vitamins, vitamins A, D, E, and K. 5. Excretion of cholesterol, as the synthesis of bile acids is one of the predominant mechanisms for the excretion of excess cholesterol. 6. Bile salts also act as bactericides, destroying many of the microbes that may be present in the food. 7. Bile salts exert a mild laxative effect by stimulating the peristalsis. 8.3 Digestion of carbohydrates. More than 60% of our foods are carbohydrates. These include the following types. 1. Starch, it is obtained from plant sources like potato, rice, wheat, cereals, etc. 2. Glycogen, it is an animal starch, highly complicated glucose polymer. It is stored in the liver, muscles, and other tissues. 3. Lactose, it is obtained from milk, also called milk sugar. 4. Fructose, mainly obtained from fruits. 5. Glucose and sucrose, these are found in all hot/cold drinks, syrups, and sweet dishes. 6. Cellulose, it is the polysaccharide of a plant's cell wall, remains undigested but has many beneficial effects. It forms the main bulk of the diet. Glycosidases are the group of enzymes responsible for the hydrolytic cleavage of glycosidic bonds found in carbohydrates. Figure 8.11 Hydrolysis of a glycosidic bond present in maltose and condensation of glucose molecules. Digestion of carbohydrates by salivary alpha amylase, ptyalin, in the mouth. This enzyme is produced by salivary glands. Its optimum pH is 6.7. It is activated by chloride, Cl, ions. It acts on cooked starch and glycogen breaking alpha, 1 rightward arrow 4, bonds, converting them into maltose, a disaccharide containing 2. Figure 8.12 Digestion of carbohydrates in the gastrointestinal tract, cellulose remains undigested. Glucose molecules attached by alpha, 1 rightward arrow 4, linkage. This bond is not attacked by salivary alpha amylase. Since both starch and glycogen also contain alpha, 1 rightward arrow 6, bonds, the resulting product also contains isomaltose, a disaccharide in which 2 glucose molecules are attached by alpha, 1 rightward arrow 6, linkage. As the food remains for a short time in the mouth, digestion of starch and glycogen may be incomplete and gives a partial digestion product such as starch dextrins, amylodextrin, erythrodextrin, and acrodextrin. Therefore, digestion of starch and glycogen in the mouth gives maltose, isomaltose, and starch dextrins. Digestion of carbohydrates in the stomach, in the stomach, carbohydrate digestion stops temporarily due to the high acidity which inactivates the salivary alpha amylase. There is no enzyme to break the glycosidic bonds in gastric juice. However, HCl present in the stomach causes hydrolysis of sucrose to fructose and glucose to some extent. HCl. Sucrose fructose plus glucose. Digestion of carbohydrates by the pancreatic amylase in the small intestine. The alpha amylase enzyme is produced by the pancreas and acts in the small intestine. Its optimum pH is 7.1 and it is also activated by chloride ions. It can act on cooked and uncooked starch and hydrolyzes them into maltose and isomaltose. The final digestion of carbohydrates occurs in the small intestine and includes the action of several disaccharidases. These enzymes are secreted through and remain associated with the brush border of the intestinal mucosal cells. 1. Lactase, beta-galactosidase. It acts on lactose and breaks it into glucose and galactose. 2. Maltase, alpha-glucosidase. It acts on maltose and maltotriose to produce 2 and 3 molecules of glucose respectively. 3. Trehalase, it acts on trehalose and breaks the alpha, 1 rightward arrow 1, glycosidic bond to produce 2 molecules of glucose. Trehalose is a disaccharide of glucose found in mushrooms. 4. Sucrase, alpha-fructofuranosidase. It hydrolyzes the sucrose into glucose and fructose. 5. Alpha-dextrinase, isomaltase. It hydrolyzes alpha, 1 rightward arrow 6, linkage of isomaltose and produces 2 molecules of glucose. Cellulose remains undigested as it contains, 1 rightward arrow 4, glycosidic bonds between glucose molecules. In humans, no, 1 rightward arrow 4, glucosidase can digest such bonds. So, cellulose passes as such in the stool. Cellulose helps water retention during the passage of food along the intestine, producing larger and softer feces helpful to prevent constipation. 8.4 Absorption of carbohydrates. The end products of carbohydrate digestion are monosaccharides, glucose, galactose, and fructose. They are absorbed from the jejunum to portal veins to the liver, where fructose and galactose are transformed into glucose. 2 mechanisms are responsible for the absorption of monosaccharides. One is active transport, against concentration gradient, i.e., from low to high concentration, and the second is passive transport, by facilitated diffusion. For active transport to take place, the structure of sugar should have a hexose ring and O group at position 2 on the right side, both of which are present in glucose and galactose. Fructose, which does not contain the O group to the right at position 2, is absorbed more slowly than glucose and galactose by passive diffusion. Figure 8.13 Mechanism of absorption of nutrients. Source: Pearson Education. Inc. 2010. Mechanism of active transport of glucose. In the cell membrane of the intestinal cells, there is a mobile carrier protein called sodium-dependent glucose transporter 1, SGLT1. It transports glucose inside the cell using energy. The energy is derived from the sodium-potassium pump. The transporter has 2 separate sites, one for sodium and the other for glucose. It transports them from the intestinal lumen across the cell membrane to the cytoplasm. Then both glucose and sodium are released into the cytoplasm, allowing the carrier to return for more transport of glucose and sodium. The sodium is transported from high to low concentration, with concentration gradient, and at the same time causes the carrier to transport glucose against its concentration gradient. The Na+ is expelled outside the cell by the sodium pump, which needs ADP as a source of energy. The reaction is catalyzed by an enzyme called adenosine triphosphatase, ATPase, or Na plus K plus ATPase. Active transport is much faster than passive transport. Insulin increases the number of glucose transporters in tissues containing insulin receptors, example muscles and adipose tissue. Both glucose and galactose are absorbed by the same sodium-dependent transporter, SGLT1, and compete with each other for intestinal absorption. The absorption of galactose is faster than glucose. SGLT2 is found in renal tubules for the reabsorption of filtered glucose. Inhibitors of active transport. Iwabayan, cardiac glycoside. It inhibits adenosine triphosphatase, ATPase, necessary for hydrolysis of ADP that produces energy of sodium pump. 2-fluorazine, it inhibits the binding of sodium in the carrier protein. Passive transport, facilitated diffusion. Sugars pass with a concentration gradient, i.e., from high to low concentration. It needs no energy. It occurs through a sodium-independent monosaccharide transporter 5, GLUT5. Fructose and pentoses are absorbed by this mechanism. Glucose and galactose can also use the same transporter if the concentration gradient is favorable. Figure 8.14 Mechanism of absorption of monosaccharides, GULT, glucose transporter, SGLT1, sodium-dependent glucose transporter 1, source, Lippincot's Illustrated Review Biochemistry 7th edition. There is also sodium-independent transporter 2, GLUT2, that facilitates the transport of sugars, glucose, galactose, and fructose, out of the intestinal mucosal cell i.e., to portal circulation. In the intestine, GLUT2 is present towards the serosal surface of intestinal epithelial cells while SGLT1 and GLUT5 are present toward the luminal surface. Defects of carbohydrate digestion and absorption and other clinical aspects. Lactase deficiency, it causes lactose intolerance characterized by impaired lactose digestion. It may be congenital or acquired. The presence of lactose in the intestine causes increased osmotic pressure, so water will be drawn from the tissue, causing dehydration, into the large intestine, causing diarrhea. Increased fermentation of lactose by bacteria produces CO2 gas. This causes distension and abdominal cramps. Presently lactose intolerance is named adult-type hypolactasia or lactase non-persistence. Treatment is simply by avoiding lactose, milk. Use lactase-treated products or take lactase in pill form.
Figure 8.15 Lactose intolerance, source, Lippincot's Illustrated Review Biochemistry 7th edition.
Sucrase isomaltase complex deficiency, a rare condition, showing the signs and symptoms of lactase deficiency. It occurs early in childhood. Treatment includes dietary restriction of sucrose and enzyme replacement therapy. Monosaccharide malabsorption, this is a congenital condition in which glucose and galactose are absorbed only slowly due to a defect in the carrier mechanism. Acarbose, it is an alpha-glucosidase inhibitor, that works in the intestine, slowing down the digestion of carbohydrates and lengthening the time it takes for carbohydrates to be converted to glucose, which facilitates better post-digestive blood glucose control. Hydrogen breath test, it measures the amount of hydrogen in a person's breath. Very little hydrogen is normally detectable. However, undigested lactose or other disaccharides in the colon or small intestine are fermented by bacteria and produce various gases, including hydrogen. Exercise questions. MCQ 1. Which of the following reactions takes place during the chemical digestion of food? Dehydration, hydration, hydrolysis, oxidation, reduction. MCQ 2. Which of the following enzymes is present in saliva? Aminopeptidase, amylase, carboxypeptidase, sucrase, protease. MCQ 3. A 33-year-old woman consulted a GI clinic for a complaint of indigestion and heartburn, particularly on intake of a spicy meal. After a thorough checkup and investigation, she was found to be suffering from gastritis due to excessive secretion of pepsin. Which of the following substances is required for activation of the enzyme secreted excess in this patient? Bile salts, calcium ions, chloride ions, hydrochloric acid, lactic acid. MCQ 4. A 40-year-old man presented with complaints of severe abdominal pain and fever. His blood reports showed elevated serum amylase levels. He was provisionally diagnosed as a case of pancreatitis. Which of the following dietary components is digested by the enzyme that was found elevated in the serum of this patient? Glycogen, nucleic acids, phospholipids, proteins. Please write answers in the comments. End of Part 1 of 2 of Chapter 8. Thank you.